Hello, everyone. My name is Yingxin, and I'm currently third-year PhD student at the UC Irvine. It's a great pleasure to be here to share two of our recent work. First one is Sailor, so it's a deep generation modeling approach for analyzing and integrating single-cell ATAC-seq data. And the second one is UFOLD. It's a deep learning method for RNA secondary structure prediction. And I will start with Sailor first. And just to give a brief background, so single-cell ATAC-seq measures measures the chromatin accessibility at single-cell resolution. So if you have reads at certain regions, it means that this region of the genome is at open state. And proteins that usually regulates the genetic processes bind to open chromatin regions. And the chromatin state for each cell can be represented as a binary vector. Regions with signal we denote as 1, and otherwise we denote as 0. And the entire data set then can be like an n by m matrix with n cells and m peaks after peak polling. And the matrix is usually very high dimensional. Both m and n can go over 100,000. So it's very high dimensional. And also it's a sparse matrix. And so it's relatively impossible to directly interpret the raw data. So usually the first step for data analysis is to perform dimensional reduction to get to a lower dimension representation. And usually this lower dimensional representation is biologically more meaningful and less noisy. With a good embedding, we can then run some clustering algorithm to annotate cell types. And this cell types information would then facilitate researchers to fully utilize the power of single cells for many downstream tasks. For example, the differentially accessible regions between different types of cells. So this is to identify regulatory elements specific for each cell type. And also we can use that information to inference the cell developmental trajectory. And this is useful when to analyze like continuous changes of chromatin accessibility within cell population. And also this is useful for assessing the co-accessibility of peaks. And this is to analyze the interactions between different genomic regions. But all these tasks, first, it requires good dimensional reduction to begin with. So why learning a good embedding is a difficult task? We think partially it's due to the technical variations introduced at the sequencing stage. So here we show some statistics of two single-cell ATAC-seq datasets from same tissue. So they are both from mouse brain, but they're sequencing from different platforms. So first, UMI is a metric. UMI is a metric for measuring the read depth for each cell. So on the left, you can see read depth at UMI in XS plotted against promoter ratio. So each point represents a cell. On the right, we show the bar plots of the UMI and the promoter ratios for two platforms. So from the figures, we can see clear batch effect first. So the read depths from different sequencing platforms are of different magnitudes, and this is plotting log10 scale. And promoter ratios are different as well. And also we can see great read depth variations even within the same sample. So read depth can vary dramatically. So it's a lot more serious than usually the single-cell RNA-seq case. And also for single-cell ATAC-seq, we also have the stochastic dropout. So it's similar as a single-cell RNA-seq case. So here I'll just show two simple examples how these technical variations may affect the dimensional reduction. So first, these are UMAP visualizations of embeddings generated by a single linear method called LSI on two simulation datasets. So the left one is without is the simulation dataset without read depth variation. So essentially, each cell has the same read depth, and on the right is a dataset with read depth variation. So when there's no read depth variation, you can see LSI could distinguish this rare cell type, which takes only 1% of the total population. However, when there's redepth variation, the real cell type is overwhelmed. And here's the second example. It's about batch effect. So here we show the T-SNE visualization of embedding learned by scale. So scale is a variational autoencoder-based deep learning method, and but it doesn't have specific mechanism to deal with these confounding factors like batch effect. So this is on the cross-platform dataset we just mentioned. It's from the same tissue, the mouse brain, but sequenced with different platforms. So without specific mechanism dealing with this batch-related confounding factor, when jointly analyzing these two datasets, the clusters reported by scale are predominantly driven by the sample effect rather than the true biological states of cell. So this situation is not ideal. So with this in mind, so we are trying to develop a method that could tackle these technical effects. And our method is based on a variant of VAE. So variation autoencoder. I'll just give a brief intro here. So VAE consists of an inference model and a generative model. The inference model learns a posterior distribution of latent variable Z and is parameterized by a neural network. And the generative model samples from the latent distribution through reparameterization and then reconstructs the input data through a neural net. And the latent variable Z is usually of lower dimension and is serving as a bottleneck. So it's usually used for dimensional reduction in unsupervised settings. So similar as SCVI is the famous work for single cell RNA-seq and the scale for ATAC-seq, as I mentioned before. And according to the goal of dimensional reduction, we want the latent representation to be informative on cell type or cell state. And however, in the real world, besides the cell identity, other technical variations are also involved in this data generation process, such as redepth variation introduced in the sequencing process, batch effect, and etc. And these technical variations can be observed at the time we analyze the data, but they are not related to the intrinsic biology we are interested in. However, if we do nothing about it, it will be coupled with latent states that's representing the biology states and affecting the final result for dimensional reduction. So it's not ideal, and we call these technical variations as confounding factors. So here comes our method. So we want to decouple these confounding factors with the true biological states during our training process. And we achieve this through the idea of invariant representation learning. So the goal for invariant representation learning is for inference model to learn a latent representation Z, which is independent with the confounding factor. So intuitively, we want the invariant representation or latent representation Z reflects the intrinsic biology states of cells and does not affected by the technical variations, such as sequencing depth and batch effect. And we achieve this by adding an etc. penalty term on the mutual information between invariant representation C and the confounding factor C. And to do so, we utilize a mutual information bound derived by Moyer et al. In the NIST 2018. So here comes our overall framework. So CATAC could take single cell ATAC sequencing data from multiple sources, from multiple batches, multiple samples as input, and the read depth of each cell is converted into a log normalized library size, and batch information is denoted as binary encoding. And the objective is negative log likelihood with conditional reconstruction plus the mutual information penalty between the latent representation Z and the confounding factor C. So here are the result parts. So we first experimented CATAC on 3 simulated datasets. So we benchmarked against a simple linear method, the LSI, and also scale previously proposed deep learning method on single cell ATAC-seq. So we used 3 simulated datasets and visualized the embeddings with UMAP. The first data contains 1 real cell type along with 4 major cell types. And deep learning models can capture these real cell types However, the linear model is not able to do that. Moreover, the second data set has shallower sequencing depth, so which makes the entire representation learning task a lot harder. And as you can see from the embedding, the LSI and scale does not mix this read depth very well. So basically, this color bar indicates the read depth. You can see like within each cluster, we can observe higher to lower read depth for each cluster. But with invariant representation learning, the Sailor generates better embeddings. So you can see the read depth mix pretty well, unlike scale or LSI. And the last data set contains batch effect. So essentially, we simulated two batches and jointly analyze them together. So similarly, Sailor mixes cell from different batches, while other methods doesn't do that well. And also, Sailor preserves the real cell types very well. Since for two batches, it each contains one real cell type. And this mixing can also be quantitatively measured. So we kind of calculate the mean mutual information for the embedding generated by each method, and Sailor achieves the lowest mutual information between latent representations and confounding vectors on all these three data sets. So we also experimented on the real data sets. So we tested on the Moss Atlas data set. It contains over 80,000 cells and 40 cell types. And this data set has been previously used in a benchmark paper for all previously proposed single-cell ATAC-seq computational methods. And the SNAP ATAC is one of the leading methods, and the only method could finish processing this data set in around 11 hours. And in terms of clustering metrics, both from the embeddings and the quantitative measures, we can see that Sailor is achieving better embeddings and getting higher ARI and AMS scores. So these metrics are basically evaluating how good is the embedding is. And also, Sailor achieves the lowest mutual information between the latent representation and the embedding. So between the latent representation and the confounding factor, and this can also be observed from the visualization of read depth. So the mixing of read depth is very well for Sailor. For scale, it doesn't have specific mechanism to correct read depth, so you can observe this high to low read depth distribution within clusters. And we also use Sailor to merge two cell Moss brain data sets. So as mentioned before, this data set are generated from two platforms. To compare with scale, which I showed earlier, the invariant representation learning with Sailor merges clusters from different batches together. And we then run clustering on our embedding and visualize some gene activity score of some marker gene to validate our clustering results. Moreover, Sailor can also be used for imputing single-cell ATAC-seq data, which suffer from sparsity due to drop-off during the sequencing. And since in Sailor we have control over those confounding variables during the generative process, we can reconstruct the data free of confounding factors. So we tested this on the simulated data set. So the raw simulated data is sampled from foreground peaks of bulk ATAC-seq data. And essentially, we first sample these foreground peaks and then combine with some noise and to generate these simulated single-cell data. And the simulated data contains two batches simulated with different read depths and signal-to-noise ratio. And we then run imputation on the raw data with scale, magic, and Sailor to get the imputed data respectively, and then reduce the dimensions of the imputed data and visualize with UMAP. We can see that the data imputed by Sailor are not affected by the batch effect and preserve the real cell types quite well. And since the simulated data is generated from the bulk data, we already know the ground truth, so we can compute the die score between the imputed single cell data and the ground truth. So the die score shows how well the imputed data overlaps with ground truth. So it's and it's also a well-balanced metric. So we can see that CEDAR solves this dropout issue if we compare it against the raw data. And also, if compared with scale, CEDAR outperforms scale in terms of the score. And that's all. That's all the materials for CEDAR. I think I'll pause here a little bit to see if anyone has any questions. Okay, Julian.
I have one. Yeah.
Okay.
If that. So you make this assumption of independence of the latent space and batch effects. And there used to be some work back in the macro times where people question a bit that and thought that sometimes confounding can actually overlap with biology. Have you made some thought of that, and is it something that can happen also in your data?
Right. This could indeed be an issue if like this component is actually strongly correlated to biology. So, but like for example, in our case, so the batch effect itself, this batch effect it's clearly not related to the biology. So the only thing is that the redepth it may relate to the biology and I think here, so with the current technology of single-cell ATAC-seq, when we observe this distribution, so we can first examine the within cell type distribution and then examine the cross-cell type distribution of this redepth. Then it's probably something worth looking at. And if this within cell type distribution is far significant than the cross-cell type distribution of this confounding factor, then we can probably assume that this confounding factor is affecting the embedding instead of contributing to the biology state. Hope this answers your question.
Thank you. Any other question?
Right. If there's no more, then I'll proceed to UFOLD. So UFOLD is a deep learning project for RNA secondary structure prediction. So there are
Yingxin, there's a question from Anthony Matelier. What about considering
in the chat? Sorry.
Yes. What about considering non-observed confounding factors? Would it be possible?
Right. So I'll go back to the method slide. So this thing, like the model showed here, so it would be infeasible to have unobserved confounding factors because when we do the generative reconstruction, we have to use the information from the confounding factor to reconstruct the data. So this has to be observed at the generative stage. So the rationale behind this is like the redepth. When we first take a look at this problem, the main confounding factor from the redepth and the batch effect, these are well observed at the sequencing stage. So that's the rationale behind our method. So, but if the confounding factor is not observed, then probably this method won't work. Is there any more questions? Okay, then I'll proceed to UFOLD. So, okay, so still I'm gonna start with some very brief background. So, unlike DNA with double strand and stable, and RNAs usually come with single strand, so they tend to fold into these secondary structures on their own. The structures are critical for their functions and properties. For example, translation rates, RNA stability, microRNA binding, RNA protein binding interactions, and etc. And computationally predicting the central structures of RNA sequences is a long-standing problem. The traditional methods are model-based thermodynamics methods, and they solve this by solving the structure with dynamic programming. However, recently with large annotated RNA secondary structure databases becoming available, the learning-based methods become popular. And these methods are data-driven, supervised learning methods. So they usually tend to have a higher accuracy. And in this work, we mainly contribute to the machine learning side of this problem. So here I listed 3 recently published learning-based methods. So they share a very similar structure. Essentially, they take a natural language processing perspective on this problem by first using sequential models like LSTM or transformers to first extract features, embeddings, and then through an order concatenation, get to the content map or folding format. And however, LSTM limits the speed of the program, is relatively slow to run. And the attention mechanism of transformer takes a lot of memory. And since their focus is mainly on the language modeling, or simply due to the memory limits, they didn't get deep as a convolution step. So here in our project, we wondered, instead of modeling RNA sequences as RNA as like language sequences, can we directly convert the sequence into an image-like representation that is easy to be processed with effective computer vision models? And also models like fully convolutional networks can take data with variable sizes. And this is an important property since length of RNA sequences usually has large range. And the final output of our model would be this symmetric content map with 1 indicating pairing at this particular location and 0 otherwise. So we can use this symmetric binary matrix to represent the secondary structure of a sequence. So first, I'll introduce how we construct this image-like representation using RNA sequences. So essentially, we take the canonical product of the 1-hot embedding of the original sequences. So it's essentially the pairwise auto product of each base channel and its transpose. So taking AA pairing as an example, we take the auto product of the first column and its transpose and generate a map. Indicates all positions that this type of pairing is happening. So this map is symmetric and has the same size as our content map final prediction. And if you remember this, if you remember this content map, so this representation is very coherent to that content map also. And we generate channels for all the possible base pairing situations, which is 16. And then after this product for sequences with length L, we have a 16 by L tensor representing the original sequence. And we add an additional feature map which represents how likely each base pair interacts with each other, which other base pairs as pairing probability matrix, and this is directly adapted from the CDP fold method. And for the fully convolutional network part, we adapted UNet to take our representation as input and output the contact map prediction. And UMap consists of a sequence of convolutional and downsampling layers to derive increasingly complex representation of the input at different levels. And it's followed by a sequence of upsampling layers with copying contextual information from encoder parts. And unlike other methods using LSTM or transformers to learn long-range features from sequences, here we extract features using convolutional kernels at different levels. And as more convolutions and downsampling operations are applied, the reception field of each kernel will increase. And as we go down, we are learning features from like larger range. And all operations in UFOLD are fully convolutional. That's the input sequences can be of variable length. So we benchmark UFOLD with other methods, including learning-based method and thermodynamics method on the Leewald test set. So on the left is the result trained with RNA string line dataset and evaluated on the archive2 dataset. And the right one is trained on the BPRNA dataset and evaluated on the left-out test set. And for within family performance, UFOLD outperforms all other methods in terms of F1 score. And as a data-driven method, a learning-based method do struggle a little bit when tested with sequences from unseen families. So we have to add some synthetic sequences with synthetic labels at training time to get more robust results. And this is also the limitations for all data-driven methods, also for other like learning-based methods as well. And hopefully, as more training data are available, this situation can get better. And one more thing to notice is that our method is also able to predict structures with pseudoknots. So pseudoknots are these sequences with non-active structures. And due to some limitations of dynamic programming, some methods do not support predictions with pseudoknots. So on RA-string line test set, we evaluate if the ground truth and the final predictions contain pseudoknots respectively, and summarizes the performance of UFOLD with other methods that are capable of predicting pseudoknots. We found that the prediction from UFOLD is relatively robust on pseudoknots. And benefiting from the fully convolutional network model and the GPU acceleration, the UFOLD is faster than other learning-based and also thermodynamics methods. And although our draft is still currently under revision, our web server is up and running. So if you want to try out your sequences, you can try our web server. So we support FASTA file with multiple sequences. So it's very convenient to use. Many other web servers doesn't actually support that. And before I end, I'd like to thank my advisors, Professor Xiaohui Xie and Professor Qing Nie, and also the co-author on this project, Lai Yi, and I think he's also in the seminar as well, and also his advisor, Professor Pan. And also the CATAC project is in collaboration with Professor Yingxin Cao. And last but not least, I'd like to thank folks from the lab, Hassan, Yihua, and Ayang for their help and suggestions. And that will be all. And I'd be happy to take any questions.